Good morning, dear students. Today I am going to talk about conformation of polypeptide chains. This is my week second lecture. This is a second lecture of week second. In this lecture, I am going to talk about a structure, geometry, and folding principles in protein architecture, because protein architecture is very important for functional activities. So normally, protein has different like four main structure: primary structure, secondary motifs, tertiary folding, and quaternary structure. Myself, Mehdi Hayat Shahi, assistant professor at Interdisciplinary Brain Research Center, Faculty of Medicine, Aligarh Muslim University. The contents of today's lecture is understand the protein conformation. I am going to explain about how protein conformation is important. And then also I'm going to talk about learn structural principles. What are the structural principles? And also explore protein folding mechanism. What are the protein folding mechanisms, how they are going to fold and what kind of bonds involved. Then I'm going to discuss about structural motifs. What are the motifs, structural motifs that give the three-dimensional structure of the protein? Then talk about misfolding proteins and the function. Misfolded protein and the function means when proteins are misfolded, then what the function there? There's a normal, from normal to the abnormal function when the protein are misfolded. Then also discuss about drug designing application. Then talk about biotechnology. Uh, In this reference, I have taken the reference from biochemistry. It is from Lubert's trial, well-known book of biochemistry, and also some references I've taken from the resources which I mentioned in the web page, these slides. Whenever I have taken any references on any things from picture or any picture, uh, proteins are essential biological molecules. Means the human cannot survive without proteins. It's very essential for the life. Uh, Functions of protein totally depend on the 3D structure, three-dimensional structure. Uh, Protein are made of amino acids, which I already mentioned in my previous lectures. It's made of the amino acids. And protein form of essential function protein as a enzymes, as a catalyst of many metabolic pathways, and also like many enzymes, you can say trypsin, like glucose metabolic pathway, whatever the enzymes are involved in the body or any type of enzymes, it has the protein. And then structural support, muscles proteins, they are structural support, myoglobin and all the structural supports. Then protein involved in the transport and signaling, uh, transporting proteins, they transport different ions from outside to inside the body. All the protein are involved. Then cell signaling involvement pathways has different proteins like receptors and ligand binding receptors there. So they are very important and they are made of protein. The protein is very important because of this reason. Protein is a functional unit of life, means that life is not so means exist without protein. Even everything like we have a lot of cell membrane is also made of proteins. Means protein has a lot of involvement in there in the body. That's why the functional unit. The key concept regarding protein and conformation is a peptide bond. Already I discussed peptide bond, and it is planar in structure, in nature, and also it shows hybrid characters, means resonancing that of peptide bond, and it is like a characteristic of single and double bond resonance, and almost 40% is a double bond character of peptide bond, and it is mainly in the trans dominant structure, and angle is 180 degree. Cis is very less because of steric hindrance. Then there's a two angle formed by the peptide bond, means the peptide side, that's called torsion angle, which provide the twist or a kind of a structural conformations, and that's called phi and psi. Phi form with nitrogen and C carbon alpha, and psi form with carbon alpha and carbon. They're free, and these two angles define all chain conformation, all sort of chain, because peptide bonds is not going to be means any kind of rotation, but these bonds has rotation, that's called torsion angles. Then Ramachandran plot, which I explained in my previous lecture, the importance of the Ramachandran plot, because they define and predict the conformation of the protein, possible conformation, and also predict that which conformation is not possible. So that Ramachandran plot allowed the rotation of phi and psi angles, and that is a alpha helix ideally in minus 57 and minus 47. Minus 57 is phi angle, and then minus 47 is psi angles. Then beta is permitted, beta sheet permitted. This is one of the structural dimension of the protein. Minus 120 degree of phi and plus 135 degrees psi. Then we have alpha helix, one of the structure. It has 3.6 residue in helix per turn, and the length of that residue 3.6 is 1.5 angstrom, and it has hydrogen bond NH as I in the direction to I4 of CO, means the fourth carbon of the helix has a bond, and all side chains projected odd bind, and it develop the helix dipole. Helix dipole, there, this dipolar structure. Then we have beta sheet. It is anti-parallel and parallel, both structure dominate, and then it had H bonds between the strand, hydrogen bonds between the strand. Yeah, that's very important, because it gives the stability and also protein to dissolve in the water. That's very important for hydrogen bondings. And then rise the length of each residue there 3.5 angstroms in the beta sheet. Then the folding forces which are involved in the conformational changes or different conformation of the protein, that's hydrophobic effect, which I discussed, is a dominant in nature. Then is hydrogen bonds. Then it has electrostatic interactions, then van der Waals forces, and also disulfide bond with the help of cysteine amino acids. Well said by Anfinsen principle. Protein fold because folding is thermodynamically favored, means any process is, if thermodynamically favored, is very good for the nature and also is important. To native state is a free energy, is this case is minimum, then of course it is thermodynamically favorable nature of the confolding principle according to Anfinsen principle. Conformation and configuration, these two terms totally different each other, and they have differences. They are conformation and configuration. What is conformation and what is configuration? Protein conformation. Conformation means 3D arrangement of atom. Means there is a 3D arrangement of the atom achieved by folding of the polypeptide chain. When polypeptide chain folded, they arrange 3D arrangement, and it is dynamic and flexible. Form rotation around two bonds, which already mentioned, peptide bond is not going to be rotated or not, is free planar, but other two bonds, that's called chain folding. This phi bond, phi between N and C carbon bond, and psi is alpha carbon and carbon bond. So both has the rotation around bonds, and it is because of the rotation and the conformation the proteins become functional and adopt a specific conformation due to rotation and interaction along their backbone and the side chain. This is very important. First is the adopt conformation due to rotation of the phi and psi bond, then interaction along their backbone. backbone with side chain, R chain, the side chain. This kind of interaction gives their conformation to be important for the activity of the functional form of the protein. The differences in conformation and configuration: features changes, no bond breakage in the case of conformation, no bond break. But in configuration, there is a bond breaking. If configuration changes on protein, then bond is breaking. Flexibility: conformational changes has flexibility, but configuration changes there's no flexibility. It is fixed in nature. Examples: protein folding. It is a form. It is example of conformation. But if protein become L or D at the transition between L and D in amino acid attached with that amino acid, that it's a kind of configuration changes that, and it is fixed, and it is depend on the requirement of the bond making. Well said. Also mention structure determine the function, and that's example enzyme. In the enzymatic cases, what happened? Active site is very important, and also active site specificity is very important. Mean that enzyme has some specificity to bind the ligands, and then or to change the condition or change the enzymatic activity, it must be very specifically. On this is because of conformational changes. Uh, like example, we can take a kind of hemoglobin protein. It is also with oxygen binding protein, and that is very specifically hemoglobin protein there have conformation to bind the oxygen. If any changes in the conformation of hemoglobin, and then it is not able to bind very efficiently the oxygen. Then also some small structural changes causing major functional effect. Even some small structure changes causing functional effect. And also it is very important structure is very important to know the structure of the protein or amino acid is very important, because it's for drug designing. We can design a different drugs based to know that what kind of amino acids going to be required for the fit in the particular uh, molecules or any inhibitors. Then also we need to understand the structure, because we need to understand the disease, the different disease. Like many diseases has misfolded proteins and abnormal proteins because of the conformational changes. That's why we need to know that the structure of the protein. And also application in biotechnology, for industrial application, we can produce different types of proteins um, with the help of recombinant DNA technology. That's why biotechnology application is also important to know the structure of the proteins. A lot conformation with the help of Ramachandran concept, means Ramachandran plot give us the idea that what kind of conformation is important. And it is also important to know not all rotation is possible due to steric hindrance, and only certain value of phi and psi angles are possible for alpha beta sheets or alpha beta conformation of the proteins and other proteins, other conformations. When protein fold, it is into the lowest energy state. The energy state is the lowest when protein fold, and this folding driven by hydrophobic interactions and also hydrogen bond. And already I mentioned the misfolding causing the disease. It has very clinical importance, like examples which I'm going to explain in next slide, in my coming slide. That's important, like misfolding protein, many well-known disease, Alzheimer disease. It is neurodegenerative disease, and it is for the old and aged patient. This is very common these days, and it is due to misfolding of the protein alpha beta amyloid proteins, Alzheimer disease. Then Parkinson's disease is also a kind of neurodegenerative disease, that is motor disorders, and this is also because of the misfolding of the protein synuclein. Then prion disease is also because of misfolding of the proteins. which I explained. These are the disease that is very clinical importance. That's why we need to understand the mechanism and how protein are folded, what the importance in for the life. Protein are linear polymers of amino acids as a primary structure, and amino acids linked with peptide bonds form a polypeptide chains, and each unit called residue and side chain is variable and that determine the function. Major type of polypeptide chain conformations. Alpha helix, one of the type of polypeptide chain conformation. It is right-handed coil. It's right-handed coils, and it is stabilized by hydrogen bondings. And every 3.6 amino acids means one turn. If suppose you consider this one turn, this is a 3.6 amino acid in the one turn of the helic. Uh, Common in this alpha helical protein is very common in the membrane proteins, most of the membrane. And one of the example here is keratin. And you see the backbone has the hydrogen bonding. This is a hydrogen bonding. Green is a hydrogen bondings. That's give you very stability of the alpha helical structure. Then other one is of conformation is beta sheet. It is extended strand connected side by side. Is extended connection. See, this is very extended that. It can be parallel or anti-parallel. Both features is available, and it is stabilized by hydrogen bond between the strand. It's stabilized by the hydrogen bonds. Then we have turns and loops like beta turns, hairpin turns, and reverse turns, the three types mainly. And then we have loops that is called omega loops. Uh, this is the example of beta turn. This is a beta turn. And this indicate hydrogen bonding with one, then second CO group, and the third group. This is a hydrogen. This is because of turning, hydrogen bonding. N bind with the carbon group there, one I direction, then one, then two, and third. This is a hydrogen binding direction, a number of carbon with nitrogen attached. Alpha helix and beta helix. The role of the turn and loop, it attached connect the alpha helix with beta sheet. And it often forms the active binding side, means mainly these kind of turns and loops mainly present in the active binding sides. And this is the loops. You can see it attached the beta sheet. This is a beta sheet, and this is a loop attached the beta sheets. So these are the help conformational forms of the proteins, turns and loops. And often it is available in the active and binding side of the proteins. Then we have like super secondary structure, and that is called motif, and it is combination of secondary structure. Like we have beta alpha beta motif, beta alpha motif. Then we have helix turn helix, and this helix turn helix mainly bind to the DNA. DNA for a kind of process of transcription and all DNA reason. Helix, this is the helix, then we have a turn, then helix, this is helix turn helix. Then we have Greek K motif and also zinc finger proteins. This finger proteins has also like helix, and then have zinc finger has like zinc component attached with the finger like that. This is also many transcription factors are zinc finger proteins. Levels of protein structure. Polypeptide conformation contribute at higher level, and then first is a primary structure, that is amino acid sequence. Then secondary structure is alpha helix and beta sheets. Then the tertiary structure is the full 3D foldings. Then quaternary structure is multiple chains together as a functional, the quaternary structures. Now the clinical and research relevance. Uh, First, protein misfolding diseases. There are many diseases causes by protein misfolding. One is very common disease these days in the elderly patient, old age people, and it is called Alzheimer's or Alzheimer disease or dementia. There is a loss of memory, and it's occurs after 65 years of age. It is slow, progressive disease. Onset means sometime reported after 50 or 60, but it's very progressive after 65 to 75 and very critical when the person reached the 80 years or something like more than 80 years old. So what happened in this Alzheimer disease? Alpha beta peptides and tau proteins, they are aggregate, means they are misfolded or not very functional, some sort of misfolding, and then they accumulate in the neurons. This is the normal neuron cells, and this is the brain. Half brain is a normal looks. See, there's nothing, no changes in this case, and this is normal neuron. But when people become aged, then there is accumulation of the amyloid plaque in the neuron cells, and the neuron cells then degenerate, not possible to regenerate, they are going to die. And then there's shrinkage, this is a shrinkage in the brains, and this causing the dementia, loss of the memory. Person is not able to remember their past, and also certain daily activities also they forget. So these are the amyloid, the misfolding characteristics. Then another degenerative neuro disease that's called Parkinson disease is a motor disorder. It is, in this case also there is a alpha synuclein forms of the protein that's called Levy body. Synuclein forms, they form beta sheet rich aggregate. You can see this is the Levy body, is neuron cells in the cell body. This is a nucleus. So what happen? They form as a Levy body that's called beta rich aggregate, and because of that reason, the neurons disturb, and they're very motor order disorders. And then there's a lot of muscles movement, very fast movement, because of kind of shaky disease of this Parkinson's, because of these reasons. Then another disease, that is Proin disease. It is because of Proin protein accumulation, alpha rich to beta rich is misfolding reason and cause Proin disease. Then another disease is type 2 diabetes, that is very common these days, is a metabolic disorders, and it is because of isolate amyloid polypeptide aggregation within the pancreatic beta cells. And this is the pancreatic beta cells. So what happened? It is producing the IAPP, that is isolate amyloid polypeptide. But what happened? This become fibrils, means accumulated in that, and it's called deposition of amyloid and beta cells death. Because of accumulations of this isolate amyloid polypeptide, there's a depletions or death of the beta cells, then they're not possible to release the insulin. So this is the reasons they accumulate and they develop diabetes 2 diabetes, type 2 diabetes. Then we have clinical research relevance for a drug designing application. It is very important because when we understand the folding of the protein, we are able to know the specific reason, and then we can design a drugs, again any receptors or any transcription factors for a therapeutic approach for different diseases, especially in the cancers and other disease also. Uh, then this also help in the protease inhibitors, for HIV and HCV targets active formations. So with the help of protease inhibitor, this protease inhibitor, so what happened when the viral infected cells, then they release in a maturation. So this is inhibited by a protease inhibitor. When we design a protease inhibitor against the HIV virus, so what happened? The virus is going to be die. They're not going to be mature, not be stop the maturations, and they cannot transfect or infect the further cells. So this protease inhibitors is also important. So we can target these proteases of the enzymes, protease enzyme of the virus, and then we define this inhibitor, that's called protease inhibitor, to block the function of proteases enzymes here, to not allow the viruses to become mature, to release from the cells, to further infect other cells. So that's HIV protease inhibitor, a class of antiviral medications that is very important. Antiviral medication that blocks the protease enzymes and preventing the virus from maturing and making copies of itself. This is the drug design application of protein. Then also we apply antibody design to exploit the a specific loop conformation region, CDR regions, CDR region, that is complementary determining regions of the antibody. This is going to help to design different types of antibodies. Accordingly, that custom design antibodies for different receptors. And we can also target many diseases that, like these days, we have antibody-based therapeutic conditions that we use. And then it's also important to know how we can design, and that's why complementary reason to exploit uh, for the antibodies. Then another, we have AlphaFold 21, that is the apps that revolutionize the structure prediction from the sequences. This AlphaFold 221 is going to be predict the structure. When we provide the amino acid, it can predict the structure of the that proteins is a functional, and also predict that where you're going to target, which specifically bind to the particular receptors or molecules. So this AlphaFold is an AI system, artificial system developed by Google DeepMind and European Molecular Biology that predict the protein's 3D structure directly from its amino acid sequences. In this case, you provide amino acid sequences to this system, AI system. Then what it does is genetic search and embedded means get the information. Then it search a structured database and finally give very predict the conformational structure of a protein. This is very high confidence, and this will a kind of a structure. We can, with the help of this, you can determine the structure of the folded proteins from the amino acid protein sequences, amino acid sequences. Then clinical biotechnology used. What are the biotechnology use of this protein? Is engineered disulfide improves the thermostability of industrial enzymes. With the help of disulfides bond, when we introduce a cysteine in a different enzyme with engineer enzymes, then that we use as industrial enzymes very stability, shows very thermal stable industrial enzymes. So we can apply in this case. Also, there's a de novo protein design by the Baker Labs custom fold for therapeutics. That's also a design a different folded protein for therapeutic application. Then there is an intrinsically disordered protein. There's no fixed conformation and functions. Also, we can design with the help of biotechnology tools. Then there is a coil motif, and this used in self-assembly biomaterials. Means we also use proteins in a biomaterials with different folding structure. And this is coil motif. This is coil motif. It is ubiquitous and is a very supercoil protein structure composed of 2 to 7 alpha helix, mostly alpha helix, that bond wrapped around one another. This is alpha helix coil motif, and we can use this as for nanostructure. This nanostructure as a formulated condition for a drug delivery or nanotechnology applications. We can deliver drugs inside the cells, and other function we can also use. And also we can target the antibodies in this case. It is widely used for synthetic biology as a molecular zippo, to direct the predictable bottom self-assembly as a functional as a hydrogel. Also, it works as a hydrogel, and also it help in drug delivery nanotubes. We can use for drug delivery nanotubes, and also we can use for a nanoparticle habits, mean that we can attach some molecules or drugs with this coil motif and deliver the drugs, and also target different disease we can target, or we can also release inside the cells with the help of this coil structure. We can also release that. So this is very significant as a tools, as a biomaterials we use for different applications. And also it forms nano cage. This is the nano cage like that. It is like makes surrounded by that and becomes nano cage, and in this cage we can put different molecules, or even we can put a drug to deliver inside the cells to target particular disease. So a lot of applications are based on biotechnology tools. We can develop this molecular biology with this molecules, protein molecules as a macromolecules, to useful for the very functional and also as a therapeutic target for many disease. We can use this coil motifs and of the protein. And this is the summary of my lecture. Proteins are polymers made of amino acids, and this linked by polypeptide bonds, and peptide bond formation by dehydration synthesis. And it has both N terminals, C terminals, and peptide bond. This is a peptide bond. And this polypeptide is unfolded, and also the primary structure then is folding, this folded to different structure, the folding different forms like a folded global protein, and the different process involved in the folding. This is the alpha helical structure, then we have alpha and beta structure, and this has intramolecular forces driving folding, that is the hydrophobic interactions, then hydrogen bond, then disulfide bond, then ionic bond, then van der Waals forces. So these forces are and also disulfide bond. So these forces are important for giving the folded structure or a folded global protein that's become functional. And now this folded structure give different conformation like enzyme catalyst. This is a substrate, and this is the enzyme specific reasons. And then they have molecular transport, help in the molecular transport reasons. Then also they have receptors, different receptor for the cell signaling. This is a binding sites, and this is a signal molecules. Then also has enzyme catalyst. This is a substrate, and this is the binding regions, and this is active sites. That's very specific, which has a loops and all. So these are the function of the protein, folded global protein, for different activities of the life. Thank you so much for my lectures.